welcome to a new video class. Here I like to explain the topic cell fractionation. First of all, we want to know about the cell structure of eukaryotic cell and the cell structure of prokaryotic cell. We know that eukaryotic cells and the prokaryotic cell contain different kinds of organelles. Let us take an example of eukaryotic cell. When we take the case of eukaryotic cell, we can see nucleus, cell organelles like mitochondria, chloroplast, endoplasmic reticulum, Golgi bodies, lysosome, vacuoles, and we can see lot of cell organelles. And we know that each cell organelle possesses a definite function and a definite structure. For example, if you take the case of mitochondria, that mitochondria usually occurs in sausage shape, like an approximately the shape of a burger. And we can see the function that mitochondria act as the powerhouse of the cell because they can able to generate ATP through electron transport system. So these are all the discoveries made by different scientists. So how mitochondria perform ATP, the production of ATP synthesis, or how we can able to find out the proper structure, proper shape, proper function of mitochondria, chloroplast, endoplasmic reticulum. These are all these can be possible with the help of a special technique. That special technique is called cell fractionation. In simple terms, we can say that cell fractionation is the process of extracting a cell or tissues followed by destructing the cell wall and cell membrane of such kind of cells and make into homogeneate. And after that homogenization process, we can able to centrifuge that homogeneate. Thereby, we can separate all kinds of organelles. That is the concept behind the cell fractionation. So, in simple terms, we can say that it is the process of it is a collection of procedure consisting of the extraction of cell or tissues, then followed by destructing the cell wall and cell membrane of that cells for making the cytoplasmic organelle or cytoplasmic solution into a homogeneous solution. Then after that process, that homogenized solution undergoes centrifugation in order to separate different kinds of cell organelles. That is the process working behind cell fractionation. In simple terms, that is the concept of fractionation. And this cell fractionation mainly consisting of three stages. One is called extraction, and second one is called homogenization, and third one is called centrifugation. So let us move into the first, that is extraction. Extraction indicates that our aim or the aim of cell fractionation is to separate all the different kinds of cell organelles present in the cytoplasm of a cell. We need to study the function and structure of all cell organelles which are found within the cytoplasm of cell. So, in order to study the function and the structure of all those cell organelles, we need to separate all those cell organelles. Then only we can able to study its structure function. So, for that purpose, we are depending a special process called cell fractionation. So, that fractionation consisting of three stages. One is called extraction. During extraction, we kept the cells or tissue in a solution having a particular pH and salt content. Usually we can take sucrose solution, 0.1 molar sucrose solution as the solution for the process of extraction. So in extraction we are taking a tissue or cell in order to isolate or in order to separate the cell organelle present in that cell. And second step is the crucial step, that is called homogenization. Mainly the term homogenization means If we have different kinds of solution, suppose if we have three type of solution in our hand, that three kind of solution are immiscible. It never mix with each other. We need to combine that three solution and make into a mixable solution. So that is the concept behind homogenization. So here in this scenario here, homogenization means the isolated cell, the cell wall and cell membrane of isolated cell slowly destruct, leads to the release of cytoplasmic cell organelles. So here homogenization indicates that we are making the cytoplasm of the cell into a homogeneous solution. We know that cytoplasm is a solution which is not a homogenized solution. So we are making that cytoplasm into an homogenized solution by breaking the cell wall and cell membrane. And we can make that process by different method. One is called grinding method. You can take a cell having definite cell membrane and cell wall, or you can take tissues. So during grinding, we can follow two methods. One is called manual grinding, and second one is called mechanical grinding. In manual grinding, we are destructing the cell membrane and the cell wall of cell to release the cytoplasmic content, that is all the cytoplasm including the cell organelle, by using pestle and mortar. By using pestle and mortar we can grind. So that is called manual. Whereas in mechanical we can depend machines or equipments for the grinding process. For example, we can use Porter homogenizer. We can use Porter homogenizer as a mechanical grind. It's an example for a mechanical grinder. We can depend this Porter homogenizer to grind the cell for destructing the cell wall and cell membrane. That leads to the release of cytoplasm, including the different kind of cell organelle. That is the concept behind. And we can also depend on another kind method, that is called French pressure press. French pressure press. Its mechanism is very simple. Look here, you can see a structure of a French pressure press. In this diagram, you can see the cell which is going to disrupt its cell wall and cell membrane. This is the cell. We need to disrupt its cell wall and cell membrane. So we load the cells in this vessel. And you can see a piston over here. And you can also see a narrow tube, very narrow tube over here. By using, if you use this piston into the bottom portion, if you press this piston into a bottom portion, they create a pressure. That pressure pushes the cells through this narrow tube. While the cells are moving through this narrow tube, that cell, the pressure create over that cells. So due to the pressure on the cell wall and cell membrane of that cell, those cell which is going through this narrow tube, the cell wall and cell membrane of the cell which is going through this narrow tube gets totally distant. So by this process we can be able to disrupt the cell wall and cell membrane by using a pressure. That is why it is called French pressure press. So the pressure is the main reason for disrupting the cell wall and cell membrane. And third one is called nitrogen bond. It is a simple mechanism. In this mechanism, we are keeping the cells. We are keeping the cells which is going to disrupt its cell wall. We are keeping the cells to disrupt its cell wall and cell membrane. So we are keeping the cell in a closed vessel. And the main features of that closed vessel is that we are keeping the cells in a closed vessel containing a solution with oxygen free nitrogen also present along with the cell. So oxygen free nitrogen, that is nitrogen in liquid form, oxygen free nitrogen is present along with this liquid suspension of this cell. And this is a closed vessel, and the internal environment present within this closed environment is a high pressure closed vessel. So after that, we are decreasing the pressure inside this closed container. So during that time, liquid nitrogen fastly bubble out from the solution suspension. That process leads to the destruction of cell wall and cell membrane of the cell. So here nitrogen acts as an agent for breaking the cell wall and cell membrane, because when we are lowering the pressure inside this closed vessel, that sudden decrease in pressure leads to the bubbling out of nitrogen from this liquid suspension, which is mainly responsible for the breakdown of cell wall and cell membrane. And next method is called osmotic shock. Osmotic shock indicates that it is a special technique to break the cell wall and cell membrane. In osmotic shock, what happens is, normally we are keeping the cell in an isotonic solution, a solution having same water potential or same osmotic pressure. So such kind of cell suspension undergoes, such kind of cells are suddenly kept in a solution having high water potential or in a solution having low osmotic potential. That leads to the rapid movement of water into the cells. That leads to the turgidity, that creates a turgor pressure and a wall pressure. Okay, so due to the entry of water molecule into the cell, the cell wall and cell membrane get burst. That is the mechanism working behind. And last one is called sonication or ultrasonic vibration. So here we are giving ultrasonic waves or ultrasonic vibration to disrupt the cell wall and cell membrane of the cell. Thereby we can release the cytoplasm along with its cytoplasmic content. So this is the mechanism which work behind the process of homogenization. So homogenization means we are disrupting the cell wall and cell membrane of the cell to release the cytoplasmic content, that is including the cell organelle, for converting it into a homogenized solution. After that, the third step, that is called centrifugation. That after the homogenization process, we are using centrifugation, or we are using a special machine called centrifuge machine to separate different kinds of cell organelle present in an homogenized cytoplasm. And we know that we already studied all the details regarding with the centrifugation, what is the basic principle of centrifugation. All those topics are deeply discussed here only. And in centrifugation, we know that particles are sedimented by using centrifugal force, and the rate of centrifugation is determined by the size of the particle, shape of the particle, density of the particle, velocity or speed of rotation, like that, the medium in which they are suspended. So different kinds of parameters are responsible for the centrifugation process, and each cell organelle have a definite sedimentation coefficient. So based on their sedimentation coefficient, they get deposited at the bottom of test tube in the form of a small pellet. So that is regarding with the centrifugation. Here I am not explaining all the details regarding with centrifugation. We already studied what is centrifugation, what are the different kinds of centrifugation, what are the principles of centrifugation. All those things are deeply discussed here before I think before 1 or 2 weeks. That's why I'm not going to the details of this centrifugation. So this is about cell fractionation. So in simple terms, we can say that we need to study the structure and function of different cell organelles present in the cytoplasm. For that purpose, we need to separate all those cell organelles by using a special technique. That special technique is called cell fractionation. It consisting of 3 separate procedure. One is called extraction, and second one is called homogenization, and third one is called centrifugation. That's all.